Ini sudah masuk. Ini masih 5.
Saya daftarkan ya.
Siapa? Yang email yang kemarin Pak ya, saya cek dulu. Ada enggak?
Tapi ini dosen.
Ini, wes. Dan semua.
Sudah. Mau minta emailnya lagi Pak, saya singkat di note biar enggak mau minta emailnya.
Oh iya Pak. Oke.
Ilalapia.
Ya, tapi your speaker is not working.
Pak Ganjar apa dengar saya? Pak Sony apa dengar saya? Ganjar dengar saya enggak?
Dengar Pak, dengar Pak, jelas nih.
Nah, kemudian berikut.
Ada anunya enggak? Backgroundnya saya belum dikasih ya.
Iya, saya kirimkan Bapak. Ampun saya kirim, Robert.
Memiliki visi menjadi program studi terbaik dalam menghasilkan sumber daya manusia yang mampu bersaing di tingkat lokal, nasional, dan global di bidang budidaya perairan, Aquaculture UMM terus menciptakan dan mengelola sumber daya manusia melalui berbagai aktivitas. Resmi berdiri tahun 1995, kini salah satu program studi yang berada di bawah Fakultas Pertanian dan Peternakan UMM ini terus bertumbuh. Memiliki laboratorium pusat untuk melaksanakan praktikum dan penelitian, Prodi Aquaculture juga membuka kesempatan bagi mahasiswa untuk terus belajar tentang aktivitas budidaya perairan dengan membuka Fish Edu Park pada tahun 2018. Berikhtiar untuk terus mewujudkan lulusan dengan kualitas terbaik, Aquaculture UMM membekali mahasiswa dengan berbagai sertifikasi, di antaranya sertifikasi cara pembenihan ikan yang baik. Mahasiswa dilatih untuk memahami pedoman dan tata cara pengembangbiakan ikan dengan benar. Sertifikasi ini diberikan langsung oleh Kementerian Kelautan dan Perikanan Republik Indonesia. Selain itu, 2 skema sertifikasi telah disiapkan Aquaculture UMM yang bekerja sama dengan BNSP melalui Lembaga Sertifikasi Profesi UMM. 2 skema tersebut diantaranya sertifikasi pengelolaan pakan ikan dan udang, serta sertifikasi teknisi tambak udang. Punya lulusan yang berkualitas, prodi ini mengajak mahasiswa terjun langsung dalam program studi excursion dan Pekan Entrepreneurship Student FPP Fest. Prodi Aquaculture terus menjalin kerjasama dengan berbagai pihak untuk membuka jalan kesempatan pengembangan diri mahasiswa, di antaranya kerjasama dengan PT Garin Agro Sejahtera untuk membuka kelas profesional industri udang, University of Kasetsart Thailand, dan PT Kelola Mina Group. Aquaculture terus memenuhi kebutuhan mahasiswa melalui berbagai fasilitas penunjang. Aquaculture UMM siap melahirkan profesional, inovatif, mandiri, dan kompeten di bidang budidaya perikanan.
Universitas Muhammadiyah Malang UMN adalah kampus swasta yang terletak di sebuah dataran tinggi di Jawa Timur, tepatnya di Kota Malang. Kota ini dikenal sebagai kota pendidikan, kota wisata, dan kota industri. UMM berdiri tahun 1964 dan saat ini telah memiliki 3 kampus, yaitu Kampus 1, Kampus 2, dan Kampus 3. mahasiswa UMM berasal dari berbagai penjuru tanah air dan mancanegara. Mereka menempuh studi di diploma dan sarjana strata 1 di 10 fakultas, maupun di magister dan doktor. Total UMM memiliki 42 program studi. Fakultas Agama Islam, Fakultas Keguruan dan Ilmu Pendidikan, Fakultas Ilmu Sosial dan Ilmu Politik, Fakultas Hukum, Fakultas Ekonomi dan Bisnis, Fakultas Psikologi, Fakultas Pertanian dan Peternakan, Fakultas Teknik, Fakultas Kedokteran, Fakultas Ilmu Kesehatan, Direktorat Pasca Sarjana UMM. UMM memperoleh pengakuan prestasi dari berbagai pihak, diantaranya akreditasi institusi dengan nilai A dari BAN PT, Anugerah Kampus Unggul atau AKU dari Kopertis 7 Jawa Timur sebagai kampus paling unggul kategori universitas tahun 2008 hingga 2013. Dari lembaga luar negeri, UMM meraih Asian Energy Award pada tahun 2009, serta pada tahun 2010 sampai 2011 menyandang predikat bintang 2 dari QS Star World University Ranking. Kerjasama internasional UMM telah menjangkau 111 lembaga perguruan tinggi, pemerintah dan non-pemerintah dari 31 negara di dunia, di antaranya Konsorsium 22 Universitas di Australia dalam Australian Consortium for In-Country Indonesia Studies, Konsorsium 33 Universitas di Asia dan Eropa dalam program Erasmus Mundus Komisi Uni Eropa, lembaga non-pemerintah AS, Peace Corps, BGP Engineer Belanda, Akademi Al-Haramain Saudi Arabia, Korea Energy Economic Institute, dan Kenko Korea, serta dengan berbagai perwakilan negara sahabat. Pembelajaran di UMM dikontrol melalui mekanisme untuk menjamin keberlangsungan pendidikan tinggi yang berkualitas dengan fasilitas penunjang yang representatif seperti perpustakaan, masjid, student center, asrama mahasiswa, taman baca, teknologi informasi, ruang pertemuan, ruang pertunjukan, kantin, danau kampus, dan sarana olahraga. Selain itu, UMM mendirikan berbagai fasilitas seperti Hotel Kampus UMM Inn, Gedung Pertemuan UMM Dome, Toko Buku UMM Bookstore, UMM Hospital, SPBU UMM, Bengkel UMM, PLTMH atau Micro Hydro Power Plant, Apartemen Sang Surya UMM, Pusat Produksi Pertanian UMM Farm, serta Taman Rekreasi Sengkaling. Fasilitas-fasilitas itu merupakan bagian dari income center yang digunakan untuk membantu biaya operasional universitas sehingga dapat mengurangi ketergantungan dana dari mahasiswa. Sebagai unit bisnis, fasilitas tersebut juga dapat menjadi laboratorium terapan dan membangun iklim kewirausahaan. Visi UMM menuju The Real University ditempuh melalui upaya terus-menerus mengejar grafik prestasi agar terus maju seraya tetap menjaga keharmonisan kehidupan kampus.
I love UMM.
I love UMM.
Hello, Siri. Your microphone is off.
Sorry. I'm sorry for it.
How are you?
I'm fine. How are you?
Nice to see you. Very good, very good.
Welcome to University of Muhammadiyah Malang.
My pleasure.
Yeah. Okay, Safira, can we start now for him?
Anytime. I'm
Okay.
at your
Yes,
advice.
ma'am.
You
Okay.
are the boss.
Thank you for it. Safira? Hello, Safira?
Yes, ma'am.
Yeah. Can we start now?
Yes, of
Are
course.
you ready? Okay.
Yes, I'm
Okay.
ready.
Yeah.
So, thank you very much, everybody, to provide me this opportunity to give you.
I'm sorry for it. We will start with the Master of Ceremony, Safira, our student. She will.
Sure.
Yeah,
Anyway,
yeah.
I thought that I can start now.
I'm sorry. And after that, we have like opening ceremony. Here we have our former dean, Mr. David Hermawan. He will give a speech for probably around 5 minutes.
Sure.
After that, you can start.
That is completely okay.
Yeah, yeah. Okay, Safira, you can continue now.
Okay, thank you, ma'am. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam
warahmatullahi
wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay. Good morning, all. Good morning, all. Honorable Speaker, Associate Professor Fahid Hamazadeh from Adelaide University, Vice Dean of Faculty of Agriculture and Animal Science, Muhammadiyah Malang University, Mr. Dr. Haryadi, SPI, MSc, Head a Program of Aquaculture Department, Mr. Ganjar Adiwirawan Sutarjo, SPI, MP, Secretary Office of Aquaculture Department, Muhammadiyah Malang University, Mrs. Anis Zubaida, SPI, MSc, dear all aquaculture and animal science lecture, vocational teacher, and honorable all participants of this grace lecture with topic benchmarking of fish health and freshwater aquaculture challenge in era 4.0. Ladies and gentlemen, I'm Safia Rahmania, the master of ceremony today's grace lecture. I'm very happy to see you here and welcome all of you to join this class international. Today's class is about diagnostic approach in aquatic animal virology. First of all, let's pray and thank to Allah Subhanahu wa Ta'ala, who has been giving us some blessing, happiness, healthy, and mercy. So we can attend and participate in this event without any obstacles. Praise and salutation be upon to our Prophet Muhammad Sallallahu Alaihi Wasallam, the last messenger, the best figure of this universe. The person who was able to save us from Jahiliyyah era to the Islamic era, namely Islamic religion that we love. Next, we will hear speech from our lecturer who work in Ministry of Marine Affairs and Fisheries Republic of Indonesia, Mr. Dr. Insinyur David Hermawan, MP IPM. To Mr. David, time is yours.
Okay, thank you, Safira. Good morning, ladies and gentlemen, especially for Mr. Pahid Hemazadeh. Welcome in this Zoom meeting. I hope next time we can. I would like to meet with you in Adelaide. I want to come to Australia, and thank you for your coming here. And I hope you are nice here and meet with me and all participants. Especially, I would like to inform you that in this discussion, also have participants from the company in Indonesia, and welcome to Miss Henny Budi Utari PEC from Charon Popan company, and then Mr. Budi Setio from the government fisheries, local fisheries in province of East Java also come here and attend with us to be a participation, and also the teacher from all Indonesia high nursery, especially in the fisheries, and of course a student and the lecture from UMM, especially from Department of Fisheries. And I would like to thank to Miss Dwi Hastuti. We can do about this discussion, and I hope this discussion to be a learning for us, especially in how to prevent, how to diagnose in the virus in fishery of especially in the stream of Indonesia. You know that in the world that disease from virus is very dangerous, and I think the experience of Mr. Professor of Mr. Farhid, I can tell about an information and explain, of course, for the viruses in Australia, also in Indonesia, maybe in the next. Thank you very much for your coming. I am happy. I hope the next time we can meet with Mr. Farhid in Adelaide, and welcome to UMM. I hope you are coming here, and good luck, and thank you very much for all participants and all audience in this meeting. I hope what will be talk what Mr. Farhid Professor we can will be useful for us, especially for the student, a teacher, and also the company in Indonesia. Thank you very much. The time to be yours to Mr. Safira. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay, thank you, Mr. David, for the speech. And next, we will announce about our speaker today. Our speaker today is Associate Professor Farhid Hemazadeh. He is one of lecturer in University of Adelaide, Australia. And the moderator is Mrs. Sri Dewi Hastuti, SPIM Aqua. Now, I will read a curriculum variety of Professor Farid Hemazadeh.
Yes, it is my pleasure.
No. School of Animal and Veterinary Science, Adelaide University, Australia. Now, he is Associate Professor in Virology and Coordinator of Immunology and Infectious Disease. The research project from him is investigation of viral and bacterial pathogen in bovine respiratory disease in Australia. Molecular and serological investigation of canine parvovirus in Australia. Molecular epidemiology of koala retrovirus and chlamydia in South Australia. And before the presentation starts, let me tell you how the presentation will go. First, we will invite our moderator and then let Professor Farid Hemazadeh to present the material. After that, there will be a question and answer session followed by a conclusion. And don't forget to all participants to fill the form for questions. Now, we come to our main program. We will be led by our moderator, Mrs. Sri Dewi Hastuti, SPIM Aqua. To Mrs. Sri Dewi Hastuti, time is yours.
Your microphone.
I'm sorry. Yeah, thank you, Safira. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Welcome to today's guest lecture. This is the second series of international guest lecture organized by Fisheries Department, University of Muhammadiyah Malang. I'm Sri Dewi Astuti, the moderator of today's lecture. I'm very pleased and excited to have all of you here and welcome all of you to this guest lecture. Our speaker today is Professor Farid Hemazadeh from School of Animal and Veterinary Sciences, University of Australia, University of Adelaide, Australia. Good afternoon, Professor Farid.
Yeah.
Yeah, I hope you have a wonderful afternoon.
Yeah.
How's the weather today in Adelaide? Sunny
Yeah,
day?
it is a very nice day today.
Okay.
Yeah, actually it is very pleasant and a little bit unusual. You know that the weather is in Adelaide is very changeable.
Yeah, very change. Yeah. Here in Indonesia, it's a rainy season, so almost every day we have a heavy rain and a little bit cold as well.
Yeah. Before I start, I need to tell you that probably
Yeah.
you know better than me. The Indonesia is my second home. I consider Indonesia as my second home because I dealt with the Indonesian colleagues and the friends in last at least 17 years. I come to Indonesia many times. We have the collaboration. I have the collaboration research project, at least 4 research projects with the Balitvet.
Yeah, you told me before in Adelaide.
Yeah,
I'm still remember that.
I know Bogor very well. I consider myself as a Bogor boy.
Bogor boy.
Yeah. I have also collaboration with the University of Gajah Mada for say more than 10 years. We have lots of research projects and the exchange students. Lots of students from Gajah Mada University come to my lab. Probably you saw lots of Indonesian students. In my lab, I have also collaboration with the Nusa Tenggara University. Probably you remember Putri. And lots of people from Balitvet coming and going. Ningshi, Simpson, Indro, and many others. I know just a little bit of Indonesian, and I would like to start with selamat pagi to everyone. Say first to you, sorry, to organize this event for me. It is my pleasure, my deep heart pleasure to be as a guest lecturer in your university. And also thanks to Dr. David for the introduction. It is my pleasure, and I love to host your students in my lab while I have very good experiences and very good memory from all of my Indonesian students. They are excellent. They are super. You know, I would like to have more and more and more.
Sure.
For example, the very last one is Putri. Putri is still working with me. She has finished by now. Putri published 2 papers, one in Nature.
It's with ND disease as well.
With Newcastle disease virus. Anyway, if you would like, I can officially start my talk by sharing the screen. I don't know.
Before you start, I will inform the participants the rule of the guest lecture today. Is it okay for you?
Yeah.
Okay. Sorry to interrupt you. And after your explanation, we also hope, we really hope that in the coming year, we also have collaboration with you, especially our University of Adelaide, especially a School of Animal and Veterinary Science. We have also many lecture here still in master degree. So I really hope that 1 or 2 of them will pursue their PhD in University of Adelaide as well. And I really promote them that you are a very best supervisor in Adelaide University. Unfortunately, you are not my supervisor. If I know you before, maybe I will choose you to become my supervisor.
Yes, that is my pleasure.
Okay, Farhid, before we start your presentation, I would like to inform the participants about the rules of the lecture today. Firstly, please use the virtual background that have been shared. And secondly, please turn on your camera for all the participants. And thirdly, please mute the microphone during the presentation. And finally, if you have a question during the presentation or during the lecture, you can simply type the question in the chat box. Okay, thank you. So by saying Bismillahirrahmanirrahim, let's start our lecture today. Professor Farhid, the time is yours.
Thank you very much. Before I start, I need to tell you that I probably need to finish at sharp at 3:45 because I have organized another meeting at sharp 3:45. It doesn't matter because, you know, the program is there and I have enough time for the question and answers and complete my lectures. Anyway, and the second one is the break. Whenever that you feel that we need to have a break, just let me know. You can interrupt me anytime for the break, and after the break we will go ahead. I roughly divided my talk in 2 different parts. One part is very general for the general approaches in the diagnostic with and I put say few case studies mostly related to the aquaculture diseases in Australia. I put say 3-4 case studies, and I'll go through the case studies to see what was the diagnostic approaches for that, and then I'll go to like a very basic information on the diagnostic techniques that we use in here for it will be very general, but I'll try to make them say more accommodated into the aquaculture area. If it is okay with you, I can start sharing the screen by my presentation. Feel free to stop me anytime by any questions, or if you feel that the chat box is okay, we can through the chat box. I'll really appreciate you and all the other organizers, and I'll say welcome to all of those. I can see we have something around 178 participants, which is great, which is super for such a kind of the lectures. And I'm say very happy to provide those lectures for the Muhammadiyah Malang University in Indonesia. Actually, it is my first experience to give the lectures in for aquaculture people. I have some research projects in aquaculture. You know, I'm mostly the virologist, and I can consider myself as a chicken virologist. I worked with the chicken viruses for many years. But you know, you might have some questions that I don't know, because I'm not super familiar with the field of the aquaculture. Yeah, we have some research projects for the infectious diseases, for viruses. I don't know, IH and IP and the others. But you know, if you come to the physiology or the anatomy or the very specific pathology of that, are totally illiterated. So anyway, I can start sharing the screen.
Yeah.
So do you see the screen?
Yeah.
Very good. I'll do the full screen and go ahead.
Okay.
You can find my email address in here, and I'll go for the title of the lecture on diagnostic approaches in aquatic viral pathogens. I took those information sometimes from the WHO and sometimes from the OIE websites, that regularly 60% of the diseases in human and animals caused by the viruses. And I provide a list of the important diseases. Mostly, I adopted that list from the important diseases in Australia, that roughly 60% of them are viral disease. That's why it tells us the importance of the viral diseases, or study of the viral diseases in animals, especially in aquaculture. Diagnostic approaches for the viruses are always challenging and difficult for many reasons. And the first and the most important one is the amount of the knowledge and the skills and the infrastructure for detection and surveillance of the viral diseases in aquaculture is very limited in the world. And working with the viruses is always challenging because they are small, they are very sensitive, and lastly, the clinical signs of majority of the infectious diseases caused by viruses are not so obvious. That's why we cannot say detect them directly based on the regular audits. Only we can consider the infectious diseases when the mortality is happened. That's why we need to have different skills for the diagnostic approaches. I'll give you first some overview of what's going on in viral diseases of aquaculture, mostly in Australia. It will be applicable for different countries of the world. Here is the important diseases of the list of infectious diseases provided by OIE, and they are notifiable diseases. As you can see in here, majority of them are the viruses. So, few words about the importance of the aquaculture industry in Australia. You will see that the majority of them, 43%, are the rock lobster, abalones. Those abalones are suffered heavily in last few years because of a viral disease caused by a herpes virus. We call it abalone herpes virus. Prone diseases, of course, in many countries of the world, they have their own diseases like the white spot disease, sardine, shellfish, 12%, blue crab, 2%, and the others, almost 3% of the total aquaculture products in Australia. And every single of those items, when they affected by any virus, they can heavily affect economically. For example, for the rock lobster, you could see that the rock lobster is $86 million per year. So another things that makes the world of the aquaculture is more important to study is growing of the aquaculture. You will see in here that since 1950, the wild catch increased, but since 1974, the aquaculture started growing, and now almost one third of the fish and aquatic industry belongs to the aquaculture. Aquaculture has its own benefits and its own risks. We'll go through the benefits and the risks just a little bit later. I can give you a very clear example. When we compare aquaculture to the terrestrial animals, we will see that aquaculture is very young, contains more than 100 different new species that people found that they can grow them, and they have economical value as a food for human or the other industries. We have very limited knowledge on the diseases, but for the terrestrial animals, it is a very old industry. Very few domesticated species like the sheep, goat, cow, pig, chicken, and probably you can count more than say 10, 15. We have very extended and extensive knowledge on the diseases caused by mostly the infectious diseases in terrestrial animals. Lots of unknown diseases exist. You will see mortality, but you don't know why. But we mostly know the diseases caused by the infectious. Quality of the water, water-related diseases, and the diseases will spread very quickly with no limit, with no barrier in aquaculture. Probably you will see if you put a barrier here, like a very simple fence, the animals cannot pass. But such barriers in aquaculture doesn't exist. That's why all of the control and surveillance measures in aquaculture is different than the things that we learn from terrestrial animals. So we'll go to see what's happened. Regarding the pathogens, the knowledge is very limited, or in majority of the times, it is unknown. We could easily detect the diseases based on the clinical signs in livestock. But in aquaculture, majority of the cases, even in the regular, say, audits or the inspections, the signs are not very obvious until death. Diagnostic tools, which is we discuss about that today, are very limited in aquaculture. And in aquatic environment, like the transmission, stress, fluid, water quality, other diseases, and the interaction, and the most important things that today we're facing with that is the climate change. Like the increasing of the temperature, even 1 degree, will affect heavily on the ecosystem of the infectious diseases. We will talk about that just a little bit later when we talk about one of the study cases. When we say climate change, it is not, say, feeling hot or heavy rains or no rain or the flood or whatever. It changed every single angle of our life. In general, the climate change in our area means increasing of emerging diseases. numbers of the papers and the publications based on the interaction of the pathogen, environment, and the host increased in last few years. In livestock and in human, we know the numbers of the diseases that's happened based on the evidences that directly connected to the climate change. Even the current COVID situation is more or less related to the climate change. Probably if you go to the ecology of the COVID, you will see that the majority of those diseases are more or less related to the environment, and the environment could be directly related to the pathogen or indirectly related to the intermediate hosts or the vectors. That's why in countries like Australia, we have to have aquatic animal health programs to maintain, for surveillance, and of course, for control measure of any infectious diseases. The first of all, even aquatic animals, they have aquatic animal diseases. They have no barrier to move, but they are still restricted based on the ecology and the ecosystem of the infectious diseases. That's why the first and the most important control measure for all of the infectious diseases is applicable for aquatic animals, which is the movement restrictions. When we say movement restrictions, means inland or in coastal area, any movement that involve in human being should be controlled and should be restricted only and only by authorities. Of course, we need to have trained veterinarians to in place the regulations for the infectious diseases. Any fish death or fish kill and the disease should be investigated based on the passive and active surveillance. I don't know what's happened for some of the phones are changed when I just open it in here. Anyway, passive and active surveillances. Probably you are familiar with those terminologies and what does it mean, the passive surveillance and the active surveillance. Passive surveillance is always happen after observation of a disease in an area. For example, an unknown disease or an unusual disease happen in an area, and the government or the other authorities order to do a surveillance to see how the disease is spread, in which extent it is spread. But the active surveillance means we need to be one step ahead of the other diseases. Means we need to go to the risk areas and investigate for the diseases that we expect to see or possibly happening in that area. For example, the best example for that in our area is avian influenza. We know that the avian flu exists in the migratory birds. When we survey the migratory birds and we found a new strain is coming by the migratory birds, we can alarm the poultry industry that, hey, a new strain will come because we found it in migratory birds. We call it active surveillance. We need to have the plan for the emergency responses as well. We need to be prepared. We need to have the response plan. And of course, we need to have the training. And they are all need to be connected to the national biosecurity. For your information, I put some, say, case studies that more or less been involved in Australia, including the pilchard herpes virus, abalone virus ganglioneuritis, Pacific oyster mortality syndrome, which is again is a virus, prawn viruses, and we put say few cases in here, barramundi nodovirus, megalocytovirus, and koi herpes viruses. We put those information as a case studies because they are all involved in more or less different diagnostic approaches for detection of the viruses. So the first one was the pilchard herpes virus. It was not listed disease, means it was not notifiable disease, but it was happened in many cases in all of the southern coast of Australia and New Zealand. When the disease spread, mortality reached to up to 60% of that, and it was heavily affected not only the pilchard ecology and the ecosystem, but also the other animals that related to the pilchard as a source of their diet. For your information, the mortality was happened in like a 12,000 kilometers coastal area. There are some photos that taken by the government of South Australia, South Australia, to show the impact of the pilchard death caused by the virus. You will see that, you know, this much of the pilchard is almost 100 pilchards, and you know, you will see kilometers, millions, or even billions of the dead fish in the coastal area. What was the reason for that? As I said, it is one of the case studies that happens in government of South Australia in SARDI, South Australia Research and Development. It shows the disease at the first instance. They found that disease happens in the gills, and the gills lesions, when we see them under the histopathology, they are inflamed, and lots of fluid exists in the gills when we compare them with the normal one. So the first approach that we have in here is we have to have very good histopathology understanding of the existing species that we have in the country. For example, if someone open it and see it under the microscope, but we don't have the normal one to compare, we cannot detect any disease. So that's why histopathology plays an important role to start or to initiate the diagnostic procedures for any viral disease. That's why it goes the respiratory failure, because you know no chance for the oxygen exchange in the gills, and the animals will die because of the hypoxia. You will see the disease started in this coastal area of Australia and spread almost within 1 year or less than 1 year in all of those coastal areas. It shows many different things. First, the biosecurity measure is very hard to establish for the water transmissible viruses. And the second, we need to consider another things. We need to know, we need to have a good information on the nature of the viruses. We know that the herpes viruses cause latent infections, means when they establish in a population, they stay almost forever. We have lots of herpes viruses in our body. Roughly, for your information, say 35% of the human beings are infected with the herpes simplex virus that causes the cold sore. But you know, very little number of the people you will see that they show the signs of the herpes simplex. It tells us roughly 85% of the infected people with the virus, they are in latent infections. We have the same scenario in here. The latent infections exist, Even based on the retrospective studies that they done in the pilchards, they found that the infection exists for many, many years with no clinical signs. And what's happened? The disease suddenly spread, mostly because of the stress. And the stress is directly related to the increasing of the temperature of the seawater. Even 1 or 2 degrees, it causes different growing of the different algae and the different microorganisms, and those microorganisms or the algae, of course, and change by increasing of the temperature, the oxygen, the soluble oxygen in the water will be decreased, and all of those factors cause stress, and those stress activate the infection. The different pandemic that happens in last few years, still they kill up to 64% of the animals. Another disease that I want to explain, again, as a case study, and probably this time we will talk about the molecular biology of the molecular biology approach, is abalone herpes virus. Abalone herpes virus causes a disease like a kind of the paralytic disease in abalones. And when the abalones is just sucked to the rocks, they will paralyze, fell off, and they will die. Just for your information, abalone industry is huge, mostly in South Australia, because of the importation. If I'm not mistaken, the cost of one mature abalone is $85 that sell in the restaurants. And majority of them sell to the Japanese or another Southeast Asian, say, restaurants, and some say expensive and fancy restaurants in Australia. Say when you pay $85 just for one small abalone, it means it has a great value for the farmers and for the producers, for the sellers, for the restaurants, for the in general for the aquaculture industry. So the diseases started in Victoria and then gradually moved to the southern coast of Australia and Tasmania. At the beginning, they thought that it might be related to the say some kind of the malnutrition or the bacteria, but they couldn't find any. And then based on the very general PCR-based test that they developed first in Victoria, they found that it is a kind of the herpes virus. That's why at the beginning they call it abalone herpes virus. It has 1 strain in Victoria and 4 strains in other part of the country, mostly in Tasmania. So all of those molecular biology works, PCRs, and the sequencing revealed it is a new disease, it is a new outbreak, and they follow that based on the phylogenetics, and they found that the origin of the disease was in some imported fish food from Thailand, because they found that those fish, say fish pellet food that they imported from Thailand, they were infected with this virus. They didn't decontaminate it properly, and that's why the virus spread. So now I want to open another approach for you in here, that the molecular biology works provide this opportunity for us just not to detect a disease, but also follow up the disease in phylogenetic and the sequence analysis to learn how or even when this disease spread in that particular geographical region. Probably, if you are familiar with the bioinformatics, we have 2 different approaches in the phylogenetic analysis. We call one of them as phylogenetic, we call them relation phylogenetic and the evolution phylogenetics. I can give you an example for that. For example, when we say relation, a disease happens, for example, the best example of that is COVID. A disease happened in a country. When they analyzed it, they found that the COVID virus first originated from China. That's why we call it relation, means that virus is more related to the Chinese strains. For this particular case, they found that the abalone herpes virus type 1 that happens in Victoria first, it is more related to the Taiwanese, to the Thailand strain. When we say evolution, means this virus, that the new virus that we have is evolved from the other viruses. What does it mean? The best example for that is this example that we have in here. When we say we have 1 strain in Victoria and 4 strains in the other part of the country, mostly in Tasmania, means this virus mutates itself and evolve to the different strains. Means the origin of those 4 strains are not directly from Thailand. The origin of them are evolution, like they evolve. They evolve based on the evolution. Anyway, I opened this discussion in here to tell you that it is not only, say, positive, negative results, yes or no for the diagnostic test, the molecular tests, they have the greater value to tell us how and when those viruses evolved and spread in some countries. So again, as you can see in here, again, histopathology will work. That's the healthy abalone. You can see the pads in here. When they paralyze, those pads will be shrink. The shrinking of that is mostly because of the degeneration of the neural system. They don't have, say, nerves with the explanation that we have. But you will see that lots of, say, cellular necrosis and the I don't want to say immune cells. They call it like a kind of the hematopoietic cells. They immigrated in that area, and they cause a kind of the inflammation. That's why, again, having a good histopathology background of the healthy diseases versus the healthy tissues versus the disease tissues will tell us lots of different stories. Anyway, that's explain how the disease is spread. And based on the surveys that we have in the South Australia, we can clearly say that we are free from the abalone herpes virus. It is one of the abalone farms, and they are the wild abalones that exist in the different coastal area. Probably this one is quite big, and I believe that it worth more than $100. So for each. The Pacific oyster one, again, it is a kind of the herpes virus that caused mass mortality from 2008 to 2010. It happens mostly in Europe, and then in 2010 start in Australia and New Zealand. The origin of that, we don't know how this virus is spread, but based on the phylogenetic, it shows lots of similarities to the European strains, like the French strain and the Norwegian strain and the Italian strain of the oyster Pacific. The most likely is, say, illegal importation, or some of those oysters stick to the, say, transportation ship, and they spread the disease to Australia. Again, it's still considered as one of the biosecurity failures, but the origin of that is still unknown. Because of the importance of, at the first slide, I showed you the importance of the Pacific oyster industry in South Australia. They established a very strong restriction rules for the oyster farms and the wild catch, and based on that, they have lots of successful control programs for the oysters. I don't have like the very updated information, but it seems that in last, say, 8 years, the oyster industry is still growing, but it hasn't reached to the full capacity as before. So at the first instance, the main and the most important things that we could diagnose is existing of a kind of the inclusion bodies in the infected cells in the oysters. You know, the nervous system, the digestive system, the blood circulation system of the oysters, they are very, very basic, and they are totally different than the developed animals. That's why in majority of the cases, the histopathology signs from the other animals are not applicable to that. The only things that we could see, we could see the inclusion bodies inside of the cells. Inclusion bodies are mostly caused by the viruses, and it is a kind of the interaction between the virus and the host when they cause the inclusion bodies under the light microscopy. And under the electron microscopy, we could see the enlargement of the nuclei and migration of the chromatin. You can clearly see the chromatin migration at the nuclei in here, or you can see it in here or in there, that it caused by the virus. So just for your information, as a general, say, approach to the viral diseases, when you see inclusion body and chromatin condensation or chromatin margination, you need to think about an infectious disease. You need to think about a virus. Which virus is that? You know, it is the next step. So after seeing those signs in electron microscopy, the next step is PCR. Based on the PCR, we could say it is positive or negative, and of course, we could analyze the sequence, and the sequencing will tell us lots of stories. I'll talk about the sequences at the very end of those slides that we have. Siri, do I have still enough time to continue, or do you want to give the people break, because it is already to say 17, 18?
Yeah, you start the talk around 10 past 10.
Yeah.
So probably we can have a break around 10:50. Is it okay for you? It's oh yeah, 1 minute. Okay, 1 minute more. But if you want to have a break now, is it okay? It is okay.
I have few more slides, because I want to finish this discussion, and then we will go to others. If you give me, say, 5 more minutes, and then we will have 15 minutes break. Is this okay?
Yeah.
Very good.
Yeah, that's okay.
Anyway, I'll go a little bit faster, and I'll go to another case study after the oyster herpes virus type 1. The most important thing is for the emerging diseases, or for the recently emerged or unknown diseases, is test development. When we don't know anything about the germ, it is very hard to develop a test for that. Anyway, another challenge that we need to face with that is how to develop a test like the PCRs. One of the solutions for that is full genome sequencing. When we have the full genome sequencing of like an organism, we could easily find whatever other germs is in that, say, metagenomic information, and then we will find the pathogens, and we will develop the test based on the pathogens. But without having those information, it is almost impossible or hard to develop the tests. Anyway, I'll jump from the prone virus ecology, and then go to the again histopathology. At the first instance, we will see the enlargement of the nuclei in the cells of the prones. those under the they are quite visible and obvious under the histopathology studies, and then under the electron microscope, we could see the viruses. So those viruses cause mass mortality in the prawns. Regarding the barramundi nodovirus, is almost the entire coastal area of the Australia is infected with this virus, and you can see it causes the mass mortality in the like the very early stage of the life of the fish, and the stop growing of the fish by say heavily affecting of the fin of the fish. Again, histopathology will help. You will see great evacuation of the tissues in the diseased fish when you compare that with the normal fish. That evacuation cause mass mortality and infect the cause mass mortality. Again, that evacuation will help to understand the pathogenesis of the disease, and that at the next step we will go for development of the PCRs. Megalocytovirus happens in the mostly in ornamental fish, and it considered as one of the good, say, challenge models for the viruses that cause mass mortality in imported fish. Just for your information, the importation of the ornamental fish, like for the fish tanks, aquariums, and whatever, like not for the aquaculture, is a growing industry in Australia and in the world. And Australia importing, say, huge number of the fish, mostly from the Southeast Asia, originated from China, Thailand, Indonesia, and the other countries. In terms of the histopathology, you will see in the gills, they again they increased, and they increasing of the size of the cells means they inflamed with lots of, say, white cells, or the or the lymphatic tissue infiltration plus enlargement of the cells. Under the electron microscope, we could easily see the shape of the viruses. Means electron microscope help in different ways. If we could see the shape of the viruses, just for your information, it is very hard. It is very hard to see the viral particles in electron microscope when we have no information on the pathogenesis of the disease. Means the first and the best approach for the viral infectious diseases is understanding of the pathogenesis. Then we can understand the virology. The very last one is the koi herpes virus. The koi herpes virus is used for the biological control of the carp in Australia. Probably you know that the carp in Australia is considered as a pest. Means if you catch it, you are not allowed to return it to the river or the sea or to the water. You need to put it in ground. And because of the illegal importation or the immigration or the biosecurity failure, the carp is established in Australia, and it heavily affects the native fish by the Australian native fish. That's why finding a proper way as a control measure is important. Recently, the koi herpes virus is introduced to the carp, and it seems that it is very effective way. And again, it causes the inflammation and the necrosis in the gills of the carps, and they cause. So I finished this part of the talk, and probably I'll leave it to Siri to go for the next step of the talk. I don't know for how long do you want to give the people break? 10, 15?
10 minutes, is it okay?
I
Because
think 10 is okay because I need to finish a little bit early.
I know. Yeah. Okay. Thank you, Farhid. We have finished the first session. I'm sure that some of you have already question for Professor Farhid, but please be patient and wait until
anyway, within that 10 minutes, I'm still sitting here. I have my bottle of water in here. I can drink it and answer the questions if they have any question. Or
okay. Any question for this session? Farhid has already explained about some viral disease in Australia, and the main tools for diagnostic test is, if I'm not mistaken from the talk, is histopathology is very important, and then PCR based or molecular diagnostic tools is also important, and electron microscope to see the shape of the virus. Is it right, Farhid?
Yeah, that is completely right.
Yeah.
Yeah. Because just for information, because the histopathology needs like a very deep understanding and years of experience in comparing like the normal tissues with the disease tissues and understanding of the pathogenesis of the diseases. The histopathology is very challenging, and very little number of the expert pathologists exist in the field of aquaculture. That's why in last, say, 10 years, the word of the diagnostic in the field of aquaculture is deviated or moved to the molecular biology. And for the molecular biology, so it is a bit easier, and it is applicable when you have your PCR machine, your reagents, and the primers. You can run your screening. Of course, the accuracy of the tests are completely different. We will talk about that just a little bit in the next session. But yeah, the molecular biology is the, say, I don't want to say the gold standard, but it is one of the best approaches exist in diagnosis of the viral infections in this field.
Okay. All the participants, do you have any question for Farhid for this session, for the session? Please feel free to ask the question directly to Farhid by raise your hand. You can press the button provided in your screen, and then I will allow you to unmute the microphone. And after that, you can start to ask the question directly to Farhid. Or if you hesitate to ask question in English, is it okay? It is okay to ask in Indonesian, in Bahasa?
Sir, I could understand Bahasa.
Oh, you go, yeah.
Farhid, but it is hard for me to answer it in Bahasa. Probably because in my previous lectures in Indonesia, in different universities, when the students ask the questions, because it is in the field of the science, we have lots of common word in it. I could more or less understand it. No worry. If I didn't understand, I'll get your help.
Okay. You can ask question in Bahasa. Farhid understand Bahasa, and if you need a translator, I will translate the question for Farhid. Is there any question? Okay, I will ask Farhid if there's no question. Maybe later, maybe they still prepare the question while waiting for the question. I will ask Farhid, if you mentioned that molecular biology is very important, and it's the best tool to diagnose the disease, especially in viral disease in aquaculture or in other maybe terrestrial animal. So is it still need to do like histology, or is it enough for only molecular biology without any other tools?
That is a very good question. the most important things in any approach in infectious diseases is understanding of the disease production mechanisms, means understanding of the pathogenesis. Of course, histopathology will help us to understand the mechanisms of the disease. When we understand it, of course, we can control it better, we can prevent it better, we can survey it better, we can warn people, we can provide more and more information. Of course, having the histopathology information in hand, when we have the doing a survey or the diagnostic for an unknown or for a known viral infection is super important. Of course, yes, means histopathology still plays a crucial role in the diagnosis of the infectious diseases.
Okay, so that's not enough for only use one tools, but it's to be completed with another tools.
If you have more tools, it is much better, of course.
Okay. Okay. Okay, thank you, Farhid. Pak Sony, maybe you want to, oh yeah, Pak Sony. Pak Sony, yeah. Let me see yours, Pak Sony.
Okay, thank you very much, Miss Sri Diastuti. Hello, Mr. Fahid Hemazadeh. Good morning, India. I don't know in Australia.
It is in afternoon. It is
okay.
Yeah, 2:30.
Okay, 2:30. Okay. I think it's a very nice presentation from you, Doctor. I'm wondering about the infected of the virus. You said in many slides you present us about the histopathology, right?
Yeah.
Histopathology. My question is, which the organ or tissue can be infected first if the virus come to animalistic body?
So, if I want to explain it, I need to explain it in next few slides that I have in my next session.
Okay.
First,
okay, thank you. Okay.
Means you need to understand the pathogenesis of the virus, and one of the factors for the pathogenesis is you need to know what is the target organ for the virus. For example, for the koi herpes virus, for the pill-shared herpes virus, for the nodovirus, the target organ is gills. Means we have the necrosis and inflammation in gills. Say, my question is, for example, what is the target organ in infectious hematopoietic necrosis? It will be the hematopoietic tissues. Infectious infectious IPN, infectious pancreas necrosis.
Pancreas necrosis. EPNC.
The target will be pancreas. The pox virus, the target organ will be the skin. It totally depends on the virus. That's why you need to have a deep understanding of the disease first to be able to pick it up from the proper tissue. For your information, if the disease happens in the brain, same as some of the herpes viruses, and you take the sample from kidney, you will see nothing.
Okay.
That's why understanding of the disease production mechanisms is the first step of the diagnostic. If you have no information, it is better to collect as much as possible tissue from all possible target organs, from kidney, from the hematopoietic tissues, from skin, from gills, from liver, from gastrointestinal tract, from brain, from whatever that you think it might be the target organ for the virus.
Okay, thank you very much, Doctor.
No worries.
It's very interesting answer. Thank you very much.
So, to save the time, if you like, I can continue with the next step of the lecture.
Yeah, Oh yeah, that's okay. The second session, okay.
Yeah.
You don't need to have a break.
No. I had my my water in here.
Okay.
So if it is okay,
yeah,
I'll go for the next say next presentation, which is on mostly for the diagnostic, and I'll in this session I'll talk about the diagnostic virology. We talk about the sample management and the different diagnostic approaches. Anyway, again, I would ask you if you have any question or if you want to stop me anytime, just tell me. I'm more than happy to do that.
Okay.
So, when we say sample management, means we need to take a proper sample at the proper time with less possible risks. When we say risks, we can classify them in two different risks, or we call them biorisks. We call them biosecurity and the biosafety. What are the differences between the biosecurity and the biosafety? In very simple way, biosecurity means keep the workspace safe or do not put the others at risk. For example, if you come to your lab and take a sample from like a farm, we say fish farm, you necropsy that. In your lab, but with the same clothes or the same shoes, go to the visit to the other farm. You break the biosecurity rules. Means you transmit some of the infections from farm A to farm B while you are doing the diagnostic works. That's why, say, biosecurity means keep the workspace safe, to do not put the other people at risk by wash the hand, by follow the proper wasting procedures, proper reporting procedures, waste and the other things. And the biosafety means work safely, means do not put yourself at risk. That's why mostly for the transmissible diseases or for the diseases that might be transmissible by human or from human to the other animals, we need to wear the proper protection gears, like the masks, the gloves, or the, working under the biosafety cabinet, and many other factors. So, when you want to work in the virology lab to work with the viruses in general, it could be the animal viruses, fish viruses, or even human viruses. Those virology labs need to have some specific designated areas, like the office room, specimen reception and the processing room, serology laboratory, cell culture room, molecular diagnostic laboratory, cold room, dark room for fluorescent microscopy and egg handling, common equipment room, media prep room, washing and sterilization room, and the storage room. So they might be, say, those areas might be specified only and only for the virology, or might be the common areas for the other activities as well. For example, the storage, washing, media prep, dark room, or cold room. That's like a very common feature of a virology lab, which has the biosafety cabinet, the CO2 incubators, and it should be very clean, because the chance for the contamination and the chance for the losing of the viruses during the process for the diagnostic is very high. The viruses are very, I'll repeat it 3 times because it has a reason. They are very sensitive. Very sensitive to the environmental conditions, very sensitive to the temperature, very sensitive to the lights, irradiation, chemicals, and they compensate those sensitivities by super fast replication. means just if you compare bacterial growth with the viral growth, is one bacteria divided in two, two in four, four in eight, and they exponentially grow. But one virus, when get into one cell, after one cycle, produce hundreds or the thousands of the viruses. In the second cycle, we have billions. And in the third cycle, we have billions and the billions and the billions of virus particles. That's why viral diseases are always very infectious. We need to have some equipment as well. We need to have biosafety cabinets. We know we need to have the ordinary and CO2 incubators. We have the chicken egg incubator, -20 and -80 centigrade degree freezers, inverted light microscopy for the cell culture, fluorescent microscopy. Fluorescent microscopy is in aquaculture virus, say virology research labs, is very important to have, because the serology is not very applicable in fish, but the antigen detection is highly applicable, and having a fluorescent technology will help us to understand the spreading of the virus in different tissues. Filtration equipment and the refrigerated centrifuge and all other general equipment for the microbiology lab. One of the biosafety cabinets that we explain in here, there are biosafety cabinets class 1, class 2, and class 3. Regularly, class 1 is only and only for the clean works. It doesn't protect you, but the class 2 not only protect you, but provide like a clean environment for working with the infectious diseases. And for biosecurity level 3 microorganisms, we need to have the biosafety cabinet level 3, which is only accessible by the glove box, and means to protect yourself, protect the environment, and protect the samples that you work with. Here is one of the good examples of the biosafety level 3 cabinets in the virology lab. For your information, I believe that the biosafety level, biosafety class, biosafety cabinets in Indonesia exist in Balitvet, and they are heavily used for the infectious diseases and for the zoonotic diseases. I personally use that for many of my experimental works that we run in collaboration with the Balitvet and Gajah Mada University in Indonesia. The basic approaches for the diagnostic virology. The first one, and it is the golden standard. When we say golden standard, means based on, they say, Koch postulate, if we isolate some virus, we could keep it forever. And the isolation has another value, and which is the vaccine development. If we isolate a virus from an infectious disease, regardless of which animal, it has potentially a value for the vaccine development. That's why the virus isolation is still classified as a golden standard. So we know that the virus isolation in aquaculture or aquatic viral diseases is very challenging and very hard. But we tried sometimes, and we isolated and we stored and identify some of those aquaculture importance viruses, like infectious hematopoietic necrosis virus, which is a rhabdovirus. So direct microscopy could be the direct, like the say impression microscopy, like seeing the things, and it is mostly applicable for the parasites, or in some cases for the bacterial infections. But in terms of changing of the cellular morphology, it can be applicable for viruses as well. When we say direct microscopy, mostly for the viruses, we talk about the electron microscope. Under the electron microscope, if we see a virus, it is a very good evidence for detection of that. Histopathology examination. We talked about histopathology examination in previous case studies that we had, that it provides very valuable information for the next steps of the infection. or for the next steps of the diagnosis of the infection. For example, if we know that it is intracellular, say, inclusion body, intranuclear inclusion body, we could say that it caused by a DNA virus. And the DNA virus, in those cases, if it caused the necrosis, we could say it is a herpes virus. Means before testing of the sample in molecular biology and sequencing, we can roughly guess what could be the possible causative agent of that. That's why histopathology will help, and mostly histopathology for the diseases that we have very little information or the no information will help a lot. We can the next step, we can use the viral antigen detection, and viral antigen detection is highly applicable for the viral infections that known in different species, and say the specific antibodies, and mostly they are monoclonal antibodies, are developed for those viruses, and we can easily use them for detection in immunofluorescence or immunohistochemistry techniques. Detection of the virus-specific antibodies, because the knowledge for the immunology for the fish, like different species of the fish, is very limited, first. And the second, we have many different species of the fish, and their antibody profile are totally different with each other. That's why the antibody detection technologies in fish is not very developed. That's why I put it in here, but it is not very applicable for wide ranges of the species of fish and aquatic animals. But the antigen detection is important. And I put the viral nucleic acid detection, like the PCR-based tests, at the end, but I highlighted because it is most applicable, most common, and one of the most important tools that that we use for the surveillance, for the diagnosis, and for the in general for the health management of the aquaculture in different levels. I put the electron microscopy in here. Electron microscopy is very expensive and is very hard to manage. That's why when we say electron microscopy is a good tool, but we need to consider all of those limitations. For example, for such electron microscope that be able to have the magnification of up to 1 million times, which is suitable for the viruses, it roughly costs around $1 million, which is very hard to establish and very hard and of course very expensive to run and maintain. That's why lots of institutes in the world, even the specialized institute in virology, they do not have access to the proper electron microscopes for viruses. Yeah, we have different versions of the electron microscopes, might be useful for the lower magnification of the tissues or the parasites or the other microorganisms like the bacteria. But for the viruses, we need to have, say, up to 1 million times magnification, and they are expensive. Regarding the specimen management, if you want to have a good laboratory diagnostic for the viral infections, you have to have first good understanding of pathogenesis of the suspected agents. As we said, at the first of all, your inspection or your histopathology will tell you that, hey, this disease mostly happens in brain. This disease happens in gills. This disease happens in the kidney, or this disease happens in hematopoietic tissues. That's why you can easily differentiate it and make your diagnostic way more narrower to understand. Knowledge regarding the type and the duration of the illness. Yeah, we know that the viruses spread or viruses shed only in the acute phase of the infection. We need to understand how acute is this. For example, the first question that we could ask from the farmers is how long ago this disease has happened? And how many of your fish are died? And when was the very, very last mortality that you observed? So in those cases, we could easily understand that could we take the sample for a successful diagnostic, or it is too late? The stage of the infection, it is at the very early stage of the infection, middle of the infection, or at the very late of the step of the infection. Mostly at the very late step of the infection, we can't detect the viruses because, you know, immune response and of course the low load of the viruses will eliminate the viruses. The age of the patients, we know that in some cases, the very young and very old animals are more susceptible to the viral infections. And in the middle of the age, because of the competency of the immune response, because of the, say proper adaptation to the environmental condition, they are less sensitive to the viral infections. If you see a mortality in the, say, healthy and aged fish, you need to consider first an exotic infection, which is not been exposed to those animals before, and they just expose and they start to die because they have very naive immune system. Immunization status, vaccination is not highly applicable in aquaculture, but we need to consider that. We could ask because, you know, recently they started to use some vaccines for fish, and the similar diseases that reported in the same population or in the neighboring areas. What kind of the samples do we need to take? If you have the live animals, for example, in the farm fish, we might have this chance to catch the fish and inspect it and take the sample. And it is the best ever sample that we can take. Like for the virology, the key word for the good detection is the sample needs to be fresh as possible. Whole blood could be one sample, lymphatic tissues, or sometimes we can necropsy the animals. Sometimes if we have like the small size of the fish, we can homogenize the entire one and screen it for the wide ranges of the infectious diseases. The question that Dr. Soni asked was about the target organ. Of course, we need to know the target organ. Otherwise, we put ourselves in astray. Which target organ? The target organ in viral infections is always not, is always the tissue that show the most lesions in necropsy. That's why a systematic necropsy, understanding of the anatomy and histology of the fish is important for us. But when we deal with the dead animals, reticuloendothelial system, like in fish, mostly will be the hematopoietic system, or in some cases, some other related organs like the brain and the others. Liver could be the good sample for taking, but you know, and of course, we need to consider again target organs as well. When we say target organ or target organs, in some diseases, we might have just only one target organ, and in some others we might have multiple target organs. It is better to collect all of them based on the understanding of the disease production mechanisms. So I'll go a little bit on the detection of the sampling or detection of the target organ for sampling. For example, at the first instance, we need to understand the target organ. For that, we need to understand the pathogenesis, and we need to understand the pathogen. There are, of course, there are two connected matters, but they are totally different. Pathogenesis is the disease production mechanism in the host, and the pathogen is a germ. We need to understand both of them to be able to have a proper sampling and the proper diagnostic approach. How to necropsy and collect the samples? Of course, we need to have a good understanding of the necropsy and sample collection. Of course, in this case, we need to consider both of the biosafety and the biosecurity measures. How to preserve samples for the histology. In this case, we need to formalin fix it, and how to preserve the tissue for PCRs. And we will talk about that, how to preserve it for PCR, because I believe that in your field, you are more interested to know about how to preserve the samples for the PCRs. For the direct examination, as one of the approaches for the direct sampling is a fresh sample. The samples need to be fresh, and they need to be transported to the lab on ice. Or sometimes directly we can make the slide impression, like pressing the tissue on a slide and stain it. We can see, in some cases, we can see the inclusion bodies, we can see malformation of the tissues, and if they are pathognomonic, we can have one step toward the diagnosis. Electron microscopes will help. For histopathology, and we said we need to fix the tissue in 10% neutral buffer formalin for 24 hours, and then change it after 24 hours. For the longer term, we can keep it in 70% alcohol. And based on my experience, what is the best, packaging system for the tissue transportation is the wheel pack. We put the tissue and the formalin in the wheel pack, seal it, and we can keep it forever. We can keep it for many, many years for transportation, and it is very safe, and it takes less spaces. For the molecular diagnosis, the best sample is the fresh samples. If the transportation of the fresh sample is not possible, frozen samples, we can freeze them in -20 or -80. For the transportation, we can transport it on ice for the short time, or transport it on dry ice. If you transport the viral sample in the room temperature for, say, over 24 hours, transport or keep it, you are sure that all viruses will die, and the chance for a proper diagnosis is very low. For the DNA viruses, we can keep the samples in 95% alcohol. But for the RNA viruses, we need to keep it in triazole or RNA later. For the unknown samples, we need to take the samples for both DNA and RNA detection. Means we need from one animal, from one fish, we need to take the sample for DNA and sample for RNA isolation. For virus isolation, as we said, it is the golden standard. It is very hard, very challenging, not successful in all of the times, but still one of the best approaches for the diagnostic. We need to put it in virus transport medium, and the virus transport medium is the buffered, contains some protein for protecting of the viruses, and antimicrobial and antifungal agents to keep the samples alive. When you take the sample, you need to put it in the virus transport media, and of course, you need to transport it on ice for the short time, or in frozen condition for a longer time. Difference between the collection of the samples by the swabs or collection of the tissues. When we collect the tissues, it could be applicable for both of the DNA and RNA, like the DNA RNA isolation and or histopathology. But for the swabs, it is only applicable for the for the molecular works. Depends on the approach. You can take either swab or the tissue, but you know that if you take the tissue, it will be applicable for histopathology, DNA isolation, and RNA isolation. We have a rule for the virus isolation. Do not freeze the specimen for virus isolation at -20. When you store it in -20, the viruses during the freeze and thaw cycles, they will destroy, and the chance for virus isolation for the freeze thaw samples are very, very low. For the shorter time of the transportation, like the regular ice, like the wet ice is applicable, or in the fridge, but for longer time storage, -80 freezer or liquid nitrogen is applicable. For the transportation in frozen condition, the dry ice or liquid nitrogen is applicable. How to prepare the sample? When we come to the lab, first of all, we know that the viruses are intracellular microorganisms. We need to release them from the cells. That's why we need to homogenize them. Homogenization is the critical step, not only for the virus isolation, it is for the DNA and RNA isolation, and it is for the detection of the viruses. Means when you want to have the antigen detection, when you want to have the DNA isolation or RNA isolation, you need to have the you need to homogenize the samples or prepare the samples. For homogenization, we have the mortar and pestle homogenizer and the Tenbrook And for your information, I say innovated a method for the homogenization, which is connection of the two syringes to a three-way tab, and put the homogenizing say buffer in here and the tissue in there, and when we pass it through two different syringes, it will be homogenized. After you homogenize it, you need to centrifuge it and then pass it through the filters. The homogenate is ready for inoculation into the cell cultures and the others. The next step, I'll talk about a little bit of the antibody detection techniques. The most important one that we have it in here is ELISA. Serum neutralization test can be applicable for the viruses that we isolated. For your information, we have successfully isolated IHN, IPN, and VHS viruses in the cell cultures. That's why we could easily run the serum neutralization test to make sure that which virus is there and how the fish are protected or previously exposed to those. Anyway, Western blot, direct immunofluorescence, and complement fixation test, which is not very applicable for fish because complement doesn't exist in fish. But we can, for indirect ELISA, we have an antigen, the antibody, and we have the secondary antibody and the substrate that we could detect it. For immunocapture ELISA, we call it immunocapture ELISA. It is mostly applicable for the antigen detection. In here, we have the capture ELISA, and we put the antigen, and we put the detection antibody, and finally we detect it based on the substrate. For your information, those immunocapture ELISAs are very sensitive and highly applicable for antigen detection in the field of the viral infections in aquaculture. For serum neutralization, it is only and only applicable for the viruses that we isolated and purified and identified. It is not applicable for the newly emerged viruses. It is not applicable for the viruses that we haven't isolated them. That's why the applications of those techniques for the aquaculture viruses is very, very limited. I could tell you that, only 5 or 6 viruses are fully isolated, identified, and the virus neutralization assay is established for them, which is still, it is an excellent technique to work with, but it is not highly applicable in the field. For the serum neutralization, we have to have a proper cell culture, and when we have enough antibody, the antibodies neutralize the viruses. When we have no antibody in different dilutions, the viruses start killing the cells, and we have the cytopathic effects observed under the microscope. For the Western blotting, we can use the Western blotting for the serum, or we can use Western blotting for the antigen. I'll just leave it as it is in here and go to the Western blotting in antigen detection. For the indirect immunofluorescence, as we said, indirect immunofluorescence is applicable for both antigen detection and antibody detection. But the highest application of the fluorescent technology is for detection of the antigens. We have wide ranges of the antibodies, like the fluorescent antibodies, developed for the aquaculture viral antigens, and we could detect them. The detection of those antigens in the histopathology will help us to understand the spreading of the virus and how virus is located, in which part of the tissue or in which part of the cells, and it help us a lot to understand the pathogenesis of the infection. We have the direct and indirect, say, antibody antigen detection. If you have antigen in here and we have the specific antibody conjugate, we can detect it directly. Or if you want to increase the sensitivity of the test, we can use avidin, biotin coupled labeled antibodies to increase the sensitivity of the test. For the antigen detection techniques, of course, we have different approaches. Direct and indirect, we have already explained about those approaches in here. The best way for detection of the antigens is these days is immunohistochemistry. Immunohistochemistry will help a lot to understand the disease production mechanisms, and it has few more advantages compared to the immunofluorescence. For immunohistopathology, first, for immunohistochemistry, not immunohistopathology, for immunohistochemistry, the first and the most important things is we don't need the fluorescent microscope. We can see the slides under the regular microscopes. When we make this slide, we can stain it, and when it is stained, it will stay for very, very long time. It is again another advantage. For the fluorescent one, the fluorescence during the time will be disappeared. But for immunohistochemistry, because it is fixed, we could see it. For example, you made some slides, and the next year you want to compare those slides with a new disease that's happened. You can easily come and see and compare them. That's why immunohistochemistry is a better technology for detection of the viruses. Just for your information, I made a quick search yesterday on the available antibodies for immunohistochemistry for the aquaculture, and I found that wide ranges of the antibodies are available for virus infection, and it will for the viral infection in aquaculture, and it will help you guys when you want to detect or follow up a disease to know that those antibodies are available. You can establish the test, and you can use it as a surveillance or the diagnostic tool in your lab. Immunofluorescence is still okay to see, and the beauty of the immunofluorescence is we could see when and how the viral antigen expresses at the surface of the cells. It is intracellular, it is extracellular, it is more related to the nucleus, it is more related to the cytoplasm, and lots of other good information will provide for us. For example, those information in here will tell me that, for example, in here, we are facing with the cytoplasmic viral infection that express at the surface of the cells. And in here, the virus expressed in the cytoplasm, but not in the nucleus of the cells. It will help for understanding of disease production mechanisms and how viruses spread. As you can see here, it is the primary the antigen. Primary antibody might be directly fluorescence, as we explained it before. I do not want to spend more time. Exactly the same thing in here, but it is mostly for antigen detection approaches. The only advantage for the secondary antibody approach is it is more sensitive and it is more flexible. Just for your information, if you have the wide ranges of the antibodies in different species of the animals, if you make, say, secondary antibody conjugated, like in mouse, you can use that secondary antibody for wide ranges of the primary antibodies with no problem. The agglutination test is, again, highly applicable for the viruses that isolated and identified. For example, it is applicable for the pox viruses. We have the fish pox viruses in fish that cause the infection in the skin, or we have the herpes viruses, and it is applicable. But it is not applicable for the other viruses. That's why the applicability of the agglutination tests for the wide ranges of the fish viruses is not very high. I'll pass those. Another technology which is very old but still applicable is immunodiffusion. Immunodiffusion, the advantages of the immunodiffusion is we don't need that much of the equipment to run it. If we have only the specific antibody, for example, if you want to detect the herpes virus in a gill of a fish, you need to make a homogenate of a gill that infected with the virus, put it in one well, in here, and positive control in there. If they make a line, you will say it is positive. If they don't make a line, you say it is negative. But you know, you have to have access to the proper antibody to test it against different antigens. Because those antibodies these days are very expensive to make, and they are very expensive to use, this technique is mostly applicable for the viruses that antibodies is producing locally or available in lower price in the market. All of those antibodies that are available against the antigens are mostly applicable in immunohistochemistry or fluorescent microscopy, not for the immunodiffusion. The electron microscopy will help a lot to understand the structure of the virus, for the classification, and to see. But you know, in majority of the cases, when we make a homogenate of a tissue that infected with the virus, we can't see the virus. That's why we need to treat it in many different ways. And one of the best ways for treating of the samples for electron microscopy is immune electron microscopy. And the immune electron microscopy is, you need to mix the sample with the antibodies. Those antibodies aggregate the viruses and they put them together. When you test it under the microscope, you could see the viruses is much better than the regular one. Means the immune electron microscopy increase the sensitivity of the test by increase the chance of the aggregation of the viruses together to see them under the microscope. In here, you will see different viruses that in immune electron microscopy use antibodies, and they aggregate them together to produce the better view. I didn't see that much of the snap test, but those snap tests might be available only and only for antigen detection techniques in aquaculture. Just for your information, we recently established a technology in our lab. We call it lateral flow assay, and the lateral flow assay is coupling of the antibodies with the gold nanoparticles, and the gold nanoparticles bind to the antibodies. And when we have the antigen in place, the antigen antibody complex make like the heavy, make it heavy. And when we put it in the lateral flow membranes, it goes different lines. It is like not a very new technology. It is a technology that that used for many years in human, but application of that for the emerging viruses could be very useful, mostly for the researchers or for the vets that they work in the farm and they don't have access to the developed lab facilities. And finally, we'll go to the molecular diagnostic techniques. We will talk about the polymerase chain reaction, real-time PCR sequencing, and DNA hybridization in here. As we said, those technologies are highly applicable for the screening and for the detection. And if we use any of those PCR or real-time PCRs, the PCR products could be sent for sequencing, and those sequencing will help us to understand the phylogenetic of the viruses that we are facing with. For the polymerase chain reaction, it works based on the polymerase enzyme that amplify one strand of the viral, genome to multiple. I believe that majority of you guys, your students, your researchers, and the colleagues are well aware about the PCR technology. I don't want to spend that much of the time in here, but at the end of the day, we have millions and millions copies of that. We can visualize them under the microscope. The beauty of the PCR, the PCR is we can submit it for the sequencing, and the sequences not only confirm our tests, but also will tell us the origin of the virus, the phylogenetic similarities, and will tell us lots of different information. For example, for the avian flu outbreak that happens in Indonesia, and at the first days they thought that it is like a kind of the evolution of the Indonesian viruses that cause the flu virus more deadly for the birds. But the phylogenetic showed that the virus evolved from an avian influenza virus that reported many years ago, like a few years later, in China. And because of the migratory birds, took that virus to the different countries of Southeast Asia, and the first establishment of the virus was in Indonesia. And everything revealed right after the PCR and sequencing of the results. They showed that those sequences are more related to the Chinese strains of the virus. So for the real-time PCR, it is almost the same PCR, but rather than running the PCR product in a gel, we could use a fluorescent dye, which is the cyber green, to detect the amplification. To do that, cyber green, or the dye-based qPCR, cyber green only and only bind to the double-stranded polymerase DNA. Means if we have just a template and no amplification, we have no signal. But if we have amplified DNA, we have increasing of the signal. That's why we could see the amplification curve. And those amplification curves will tell us how much virus or how much target did we have at the beginning of the reaction. That's why we can quantify the target. For example, we could say we have this much of the virus in these samples. what is the meaning of that? I can give you like a very everyday example for that. For example, just because this technique for the first time developed to detection of the HIV in AIDS patients, they started to treat the patients with the antiviral drugs, and they didn't know when do they need to stop. That's why they developed those kind of the tests to be able to quantify the viruses that still exist in the patients. When they stop the treatment, when the virus is not detectable in the patients, means the patient is free from the virus. How long does it take? We don't know. Sometimes it takes 3 months, sometimes takes 6 months, sometimes takes 1 year. When do we need to stop? When the qPCR tell us we have no virus in the patients. That's why it is a very powerful tool to detect the viruses in the patients. As we said, I put lots of different applications for the qPCR. We can quantify the virus load. We can detect the different viral strains. We can diagnose the infectious diseases. We can different genotypes. We can detect different viruses in the food. We can even detect the mutations, and finally the genotypes. Mostly for the virology, genotyping and the virus load are the highest applicable approaches for this technique. As we said, the sequencing is developed, and the sequencing is almost based on the PCR, but we need to use, again, for each individual nucleotide, we need to use different dyes, and those different dyes provide different signals in sequencing. Anyway, at the end of the day, when we have a sequence, those sequences can be analyzed. And if you like, we can have another session for the bioinformatics, and I can explain all of the bioinformatic approaches for you to see how we can compare one sequence to the others, and how can we create a phylogenetic tree, and how those phylogenetic trees will provide information for us to see how similar or different are those microorganisms. Probably for the survey types or for the biosecurity approaches, the sequencing will help. Next generation sequencing is, again, a new technology. For the next generation sequencing, we don't need the primers. In this technology, we isolate the total DNA or total RNA. We break the DNA in very small pieces and couple those small pieces with the adapters. And the adapters are mainly random hexamers. I mean, those random primers bind randomly to the DNA, and those amplifies. And each individual adapter coupled or fused to a bead, and that bead has a specific barcode. When it is amplified, all of those barcodes finally will be read by a machine, and it provides millions and millions information for the different genes, gene arrangements, gene expression, and lots of other information. Just for your information, in a PCR based, we could detect 1 gene in uniplex, maximum 4 genes in multiplex PCRs. And we can detect it like, say, the expression or different levels of that. In next generation sequencing, we could detect at the same time up to 30,000 genes. That covers wide ranges of the host and the pathogens. Majority of the new pathogens that discovered in the last 10 years, they are all based on the next generation sequencing. Because when we don't have information on the genome, we cannot develop the PCR. Sorry. When we have no PCR, we cannot detect any pathogen. But next generation sequencing will help us to detect new pathogens, to develop the gene bank information for the other people that they want, to create the PCR PCR methods, PCR techniques, and of course it increased the knowledge of us in infectious diseases a lot. Just for information, it will say majority of the PhD in the field of the virology are mainly focused on next generation sequencing to understand the pathogenesis of the infections. For the Sanger sequencing, probably we explained that, you know, each individual nucleotide will be coupled with different colors, and each different color provide different signal. We'll talk about it. For the DNA hybridization, which is the very last things that we discuss about that, we have to develop a probe, and those probes bind to the specific DNA, and we can visualize it in the radiology films, or we can visualize it in the southern blot, and the southern blot is the detection of the DNA in the membranes. Actually, by increasing of the use of the next generation sequencing and the qPCR, the hybridization techniques are much less applicable for the infections, but you know, still they are golden standard for some of the infectious diseases that, you know, the probes are well developed for them, because they are very quickly we can detect them, and they are say much cheaper than the full genome sequencing. For your information, the cost for the full genome sequencing, only full genome, not analysis, full genome sequencing for one sample is something around $2,000. But you know, the cost for a probe could be very cheap. Say by $50, you can run hundreds of the reactions using the same probe. That's the procedure for the hybridization. I will not take that much, and probably I finish in here to provide more time for the people who want to have the questions.
Okay.
Anyway, I hope you enjoyed the talk, and I hope this presentation was useful for your audiences, because as I said, I'm not the aquaculture scientist. I mostly took the approaches for the diagnostic, for the, like to say, general virology. Thank you very much.
Okay.
Terima kasih.
Yeah, thank you very much, Professor Fahid, for giving us such information and interesting presentation. I'm sure that all you explain is very valuable for us. It's like new information for us, especially for our students. Yeah. We have now 2 questions from the chat box.
Yeah,
I'll read it for you?
I'll go through the chat box to see.
Yeah. The first one is, maybe I can summarize the question. The first one is there any disease problem caused by virus in shrimp aquaculture in Australia? And if so, what is the problem, the virus, and how Australians cope with this disease, and how to control disease in Australia? It's the first question.
Yes, we have the white spot virus in shrimp and the prawn industry in Australia. They are all. Let me stop sharing to have more. Yeah. Now I can see the chat box. The best control measure for that is providing the virus-free population for the farmers. Virus-free, say, shrimp for the farmers to grow. As you know, that it is very hard to remove a virus from the environment when it is established. The best way is removing of the infected hosts for that, for the shrimp industry. Yeah. And the next question, based on your experience, there is a method could be used to kill the viruses. Of course, we have lots of methods for killing the viruses. But you know, if I want to take the aquaculture approach, I want to share one of my very recent experiences in killing the viruses. We established a research project with a scientist, which is the scientist in the field of physics. And he established what we call it, a plasma jet technology. And we trying to establish this plasma jet technology to control the Pacific, to control the oyster herpes virus. And how is it possible? We know that the oysters are very sensitive when they are very, very small to the virus. And for the oyster farms, when they want to grow them, they take the water from the sea, filter it, put it in their hatcheries, grow the oysters, and they took the wastewater out. If the water is infected with the virus, the filtration cannot kill the virus. Means the viruses can pass through the filters. So the next step is they try to establish disinfection mechanisms by putting the UV lights. You know, they make a kind of the tunnels. When the water is passing through, they put the UV lights and the UV disinfect the water. But they found that it is very, very high energy consuming, very expensive, and it is not very effective. Why? Because the penetration of the UV in water is very limited. And it is highly affected by the particles in the water. So we developed the plasma technology, and the plasma is highly penetrating. We can penetrate the plasma, say, up to 1 meter into the water. Even we can create the plasma inside of the water. And then it is one of the most innovative water treatment technologies that we introduce, or we're trying to introduce to the industry. That's, we put the, say, plasma jet, and it is not very expensive. A plasma jet machine, and the plasma jet machine is, say 50 centimeters by 50 centimeters by 10 centimeters, only that. And it has a probe. The probe gets out to the water, and it creates the oxygen plasma. Means it is not only kill the viruses, but increase the oxygen content of that. And we found that it is very, very effective. Anyway, we're trying to introduce this technology to the oyster farmers, and they are very interested to know about that and to use it. Yes, we found some ways for killing of the viruses. They are very effective methods in the lab, but not very useful in the environment. Any more questions?
Okay. Thank you, Farhid. Is there any question? Any other question?
Ma'am, I will ask question.
Oh yeah, Safira.
Okay. Hello, Mr. Farhid. I am
Yep.
a student from Aquaculture Department. I will ask to you, we know that viruses are specific, where the tools used to detect viruses are only for those viruses. My question is, if the virus is mutated, can we use the virus detector for viruses that have mutated? Thank you.
Yes, we have the methods for detection of the mutations in the viruses. So probably if you are you familiar with how to develop the primers for the PCRs?
No.
So the thing is, how to develop the primers is, we can develop the primers for the stable genes. And the meaning of the stable genes are the genes that they don't mutate a lot, or they are more stable during during the time. And how do we know that? By comparing of different sequences of the same genes, and we can detect the stable genes. When we have the stable genes, we can develop the PCR for those, and those PCRs will work. But in some of the cases, if we develop a PCR based on the stable genes, that within those stable genes, we have the variations. Means we have two different stables at the two parts of the genes, but in the middle of that is very unstable, means mutated. So in that case, we can develop the PCR, and we have two different approaches for the genotyping or detection of the mutations. The first approach is sequencing. Now sequencing is very, very cheap and simple. For example, for sequencing of the short band, you need to pay only, say, $20, which is nothing. 200, say $10 is 10,000, 20,000 rupiah, roughly.
Oh, 20,000 murah. Yes.
No, 20, say 200,000 rupiah. About $20, roughly $20. So it is sequencing. Another way for that, which is we developed this technology in our lab, is we call it high resolution melting analysis. Actually, it was a part of my presentation today, but I removed that because I thought it is a bit complicated. For the high resolution melting analysis, we can detect the mutation of the genes within the PCR products in a very short time, and we don't need to do the sequencing. And it is based on how like a PCR product is melting in different temperatures and release the signals. Anyway, if you like, I can have another lecture for you, talk about all of those genotyping.
Okay. More question? Pak Doni, you want to ask question? Okay. Yeah, Pak Doni.
Okay, thank you, Mrs. Riviastuti. Thank you, Mr. Farhid, for your very great presentation. I have a question. Yeah, I have a question, but maybe this is a little practical. In Indonesia, Vanami shrimp is always been a major commodity from aquaculture, and until now, the cultivation continues to be developed in large scale or even for small scale, like traditional farmer with a simple technology and do not have a strict biosecurity or just low-level biosecurity. Such Vanami farmer are of course only able to take a simple precaution. Because the white spot virus has been reported to spread in all water in Indonesia. So the farmer believe the virus is already present in the pond and unavoidable, but they have to keep the cultivating and be able to compromise against the virus.
Yeah.
In your opinion, if the virus is already present in the pond, while shrimp do not have any specific or adaptive immune response, what approach might be taken by traditional farmer so they does not eliminate all productivity, or how to maintain a virus keep in the lab phase?
Yeah.
Thank you. Thank you, Mr. Farid.
That is, I call it $1 million question. It is a very hard question to answer, because for the white spot, you need to consider wide ranges of the factors, including the environmental factors and including the host-specific factors. I do not completely agree with you to say we have no say adaptive immunity in shrimp, because we had a research project on and made a kind of the what we call it, the kind of the vaccine based on the bacteriophage technology, because the bacteriophages are more stable in water. When you make a bacteriophage-based vaccine and you put it in water, it is more stable because the origin of the bacteriophages or from the water. When you put it in the water, but in controlled conditions, even they can amplify, they can get into the shrimp, they can make some immunity. It is one approach. I don't want to say it is a good approach, but it is one approach, could be. But you know, the first and the most important one is biosecurity. Having a good biosecurity measure is a key factor for preventing of every farm from the infections. I know sometimes establishment of those biosecurity measures is hard, but you know, it is what it is. Look at the countries that they successfully control it. They have biosecurity. So the other approach is that started in some European countries recently for the white spot disease is they start to make a kind of the genetically resistant shrimp to the white spot, and they have some levels of this the success for that. You know, if I want to classify them, first and the most important one, and I believe it plays more than 80% of the protection measure measure is biosecurity. 10% say genetically resistant microorganisms, and the lastly is vaccine. The role of the vaccine could be say less than 10%, but you know, if you know, as a manager, as a scientist, if you want to suggest them, the best suggestion is biosecurity.
Biosecurity.
Yeah.
Thank you. Very clear, Mr.
No worries.
Thank you.
Okay, is there still more
sorry,
questions?
for your information, I need to leave the session and join another meeting in 2 minutes.
In 2 minutes. Okay.
If it is okay.
Okay. Probably we should finish the lecture today, but we really hope that next time we can invite you again. Maybe I will email you for the time.
Sure.
And actually, we still have what's program with you in FGD, like focus group discussion, where all the lecture will try to discuss with you about the possibility our department to work with Adelaide University, especially School of Animal.
I am more than happy, because you know I planned for that, because you said the closing, and I thought the closing means my presentation is closed, and I organize another meeting with colleagues at the university. I'm more than happy to share my email address with whatever colleague or students that they have a question. I'm more than happy to answer those questions. Even I'm a little bit more than usual busy, but you know, I always try to respond the emails.
Yep.
I'm more than happy, and of course I'm happy to have another session with your university.
Yes,
it is my pleasure, and it makes me very happy when I collaborate with the Indonesian colleagues there, because I have very good memory from Indonesia.
Okay, thank you very much, Professor Farhid, for the presentation and also the discussion, and also for the participants. Thank you very much for your attention and for the very active participation in today's lecture. And then finally, let me give applause to Professor Farhid, and also for you all.
Thank you very much. Thank you very much.
Thank you very much. See you in the next series of the guest lecture. I will give the time and screen back to Safira.
Terima kasih, everybody.
Thank you very much for it. See you next.
No worries.
Yeah, see you next
See you
time.
later.
Yeah, see you.
Thank you for the moderator. Ladies and gentlemen, finally we come to the end of this class. Before closing the class, we will give a presentation.
He's already
okay. Maybe we have to take a photo session with all the participants. Please, to all participants, please to open your camera and we will take a photo together. Can you open the camera, please? and don't forget to smile, ya. And you give your best. Gaya tuh apa tuh? Oke, right. Please open the camera. A participant. Hello, can you hear me?
Senyum Pak Desdianto.
Udah agak sebagai bahasa Inggris ya.
Oh, nggak usah udah.
Terima kasih. Terima kasih.
Senyum dulu ya. Senyum dulu.
Selamat untuk Bapak Ibu ya yang
Selamat Bapak Ibu.
Di
foto
dulu ya, di foto dulu, oke? 1, 3, 2, 1. Masih satu halaman. Yang halaman selanjutnya belum di SS. Lagi Bapak-bapak, Ibu-ibu. Oke, on the next page. Open kamera lagi. 1, 2, 3. Oke. Thank you for the all participant. Ya,
absensi Mbak Fir, mungkin
ya mungkin untuk teman-teman atau bapak-bapak ibu dosen yang belum mengisi form absensi, untuk silakan mengisi form absensinya dahulu, atau akan saya share kembali untuk linknya. Silakan untuk bapak ibu dosen yang mungkin baru hadir, baru masuk, untuk mengisi link G-form yang ada di ruang chat.
Ini diagendakan kapan ya?
Oke.
Iya, orangnya sudah terlanjur keluar.
Iya.
Ah ya.
Bu Sri, Ibu.
Ya, Safira.
Ini diselesaikan saja apa bagaimana Bu untuk?
Ya, sudah selesai.
Oke. Ya, terima kasih untuk semua praktis. Ya.
Terima kasih untuk Bapak Ibu semuanya dan juga semua mahasiswa yang sudah berpartisipasi ya pada kuliah tamu ini. Insya Allah kalau ada kuliah tamu lagi, insya Allah Bapak Ibu yang kemarin PPG akan kita share lagi. Ini ada Bu Erika, Bu Nurul Mulasari. Selamat untuk semua Bapak Ibu yang lulus PPG. Ya, terima kasih.
Bapak Ibu.
Ya. Sudah Safira, monggo silakan mungkin tutup.
Oke. Ya. Thanks you to all the participants and bye-bye. See you on the next international class. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Terima kasih banyak Bapak Ibu.
Selamat, Erika.
Terima kasih Pak Doni atas bimbingannya selama ini. Keburuk.
Terima kasih Bapak Ibu dosen.
Terima kasih Bapak Ibu.
Terima kasih sudah hadir.
Terima kasih dosen.